Okay, dear audience, thank you very much for the invitation. I'm very glad to participate in this very nice conference. So a couple of years ago in our core facility, this G400 DNA sequencer were installed, and I'd like to share our comparative results using this technology. So actually, in our core facility, we use classical short-read sequencing technologies. So the Illumina Next 500 is available, and also, as I mentioned, the DNBSEQ G400, which represents a high throughput NGS platform, and the Illumina platform represents a medium throughput sequencer. So we provide various genomics and transcriptomic analysis, including whole genome sequencing, exome sequencing, and transcriptomic analysis, and various epigenome-related method. Today, I will focus on the transcriptome sequencing, and very shortly, I will talk about the single-cell analysis. So we use the stem cell differentiation system. In our laboratory, we are interested to generate immune cells and blood cells from pluripotent embryonic stem cells, and because these immune cells, especially the dendritic cells or lymphocytes, maybe in the future can be used for immunotherapy. But to accelerate this differentiation process, we probed various transcription factors to facilitate and to improve the immunogenicity of these cells. So we tested a bunch of transcription factors, and especially we found that the Runx3 and Zbtb46 strongly modify the phenotype of the blood cells. So, for instance, we found that Runx3 instructed embryonic stem cell-derived dendritic cells have an enhanced maturation capacity, immunogenicity, and T-cell activation capacity. So this is comparable with those cells which can be generated from the bone marrow-derived adult hematopoietic cells. In addition, as I have mentioned, the other factor was the Zbtb46, which has a huge impact during the early blood cell development. We found that if we overexpress this transcription factor in blood cell progenitors, the myelopoiesis, the myeloid blood cell development, and the cell differentiation is negatively impacted. In contrast, these cells tend to form erythrocytes, and they can express adult hemoglobins. So to investigate these transcription factors, we use a chemically inducible system. So we use a genetically modified cell lines. In the X chromosome, in special locus, we introduce these special transgenes together with the TET response element. So if you treat these cells with doxycycline, the reverse tetracycline transactivator will bound to this TET response element, and you can turn on your transgene. And if you remove the doxycycline, you can turn off the transgene. And we proved that with the RT-PCR that upon doxycycline treatment, the Zbtb46 transcript level was highly elevated, and even using intracellular flow cytometry, we found that these cells really express the Zbtb46 protein. But the question is, what is the genomic impact of this transcription factor? We know that we can detect a very strong phenotype. To address this question, we perform messenger RNA sequencing. So we use this Zbtb46 inducible ES embryonic stem cells or the Runx3 inducible cells. Even without differentiation, we just culture the cells for 3 days with or without induction of the transgenes. So we use 4 replicates. So together we harvested 16 samples, and we analyzed the samples with the RNA sequencing. So the very same samples were analyzed using the Illumina Next 500 sequencer and the DNBSEQ G400. So first of all, we compared the global gene expression pattern using these two technologies. As we expected, upon the transgene induction, so when the Runx3 was induced, we can see a very distinct global gene expression pattern, and this is also true for the ZBTB46. So even in undifferentiated ES cells, if you overexpress this transcription factor, the global gene expression pattern is quite different. But the major question was whether the two technology can be comparable. So the blue color represents the Illumina data, and the green one is the MGI. So you can see the pattern is quite similar. So next, we try to identify and analyze those genes which were positively or negatively regulated by the ZBTB46 or Runx3. So here you can see the ZBTB46 results. So first we separately analyze those genes which were positively or negatively regulated by the ZBTB46 with the Illumina technology or the MGI technology. Obviously, the red color represents the positive regulation. The blue is the negatively regulated genes. So we got highly overlapping gene sets. And if we put together all of the regulated genes and applying this hierarchical clustering, you can see the gene expression pattern. This is very, very similar if you compare the two technology. Even some individual samples, the gene expression pattern was slightly different, like in the second control samples, but the very same pattern were obtained using the two technology. And obviously, we also analyzed the Runx3 regulated genes. So once again, we can get a highly overlapping gene sets which were regulated, and this cluster analysis also revealed a very similar pattern. So using both technology, more or less the same genes can be regulated, and the regulation is quite similar. So in this case, we just only detected relatively few genes which show a high induction. So if we zoom in, we can see those individual genes which were upregulated upon the Runx3 overexpression. So for instance, obviously, we found that it's barely visible, but Runx3 itself was highly expressed. In addition, we found the GDF6 and the CCR7 were highly expressed, and we confirmed these results with a quantitative PCR using independent samples. In addition, we found that the member of the granzymes were highly induced. It's barely visible, but many members of the granzymes were in use. It should be mentioned that actually all members of the granzyme B family were strongly upregulated and coordinately upregulated in case of the Runx3 overexpression. So this data suggests that Runx3 probably directly regulate the granzyme gene expression. As you may know, granzymes are very important for the cytotoxic T cells and NK cells for the killing activity. And we also know that Runx3 is important for the T cell and NK cell development. So these results really suggested that the Runx3 can directly regulate this gene family. So for the study, it was carried out 1 or 2 years ago. We used the standard MPS chemistry in case of the Illuminus MGI sequencing. But this year, the hot MPS chemistry was available. I cannot also tell the exact chemical composition, but we have to update the DNBSEQ machine, and we resequence the same RNA samples using this hot MPS, and we try to compare the global gene expression pattern using the principal component analysis. So once again, in case of the Runx3 or ZBTB46 induction, the pattern is distinct, but the hot MPS and versus the standard MPS results are quite similar. Even this is a kind of aggregated analysis, so the 4 replicates analyzed together. But even if we analyze separately the individual samples, you can see the pattern. So the hot MPS and standard MPS resulted very similar. gene expression. And once again, we also selected those genes which were positively or negatively regulated. So here you can see the ZBTB46 results. And once again, the pattern is very similar and a huge number of transcripts overlapping. So we should say more or less the same results can be obtained if using the standard versus the hot MPS chemistry. And here you can see the result of the Runx3 regulated genes. So until now, I just talked about the undifferentiated cells. We also tried to differentiate the cells, the embryonic stem cells, and to analyze the effect of the Runx3 overexpression. So we generated embryo bodied for 6 days, and embryo bodies were disaggregated and further cultured at various time. And half of the samples were treated with doxycycline to induce the Runx3. Because we previously established that the both technology can get a good result. So in this case, we use the cheaper MGI DNBSEQ sequencing. And so you can see even upon the differentiation, some of the genes were positively or negatively regulated, but less genes were detected in differentiated cells. So here I just show one example. We just selected the 9-day differentiated cells, and I just show those genes which were positively regulated. So we can see that these are partially overlapped with 12 genes, and for instance, we found that once again the granzymes are highly expressed in this upon the doxycycline treatment. But interestingly, in case of the 19-day samples, we failed to detect the very same pattern. So those genes which were regulated early stage is not regulated in the late stage. It turned out at late stage the Runx3 transgene is silenced, and probably these genes are indirectly modulated by the Runx3. So in case of the differentiated cells, we obtained fewer transcripts, and the possible reason of during the differentiation, the cells were much more heterogeneous. And the other issue is that during the course of the differentiation, the Runx3 transgene induction was not so prominent. So this result prompted us to try some other technology. So that's why we also performed some single cell analysis using the 10x chromium technology. So in this case, we try to capture at the early stage the effect of the Runx3 at the early stage of the differentiation. So we harvested the cells at 8 days and 12 days. So here I just show the 8-day results. So we co-analyzed the dox-treated and non-dox-treated, so without Runx3 induction, and we identified many clusters. These clusters represent the individual cells, and some of these cells show overlapping or similar gene expression pattern. So interestingly, in case of the dox treatment, so when the Runx3 was overexpressed, we found that some unique clusters. So for instance, this cluster is not exist without doxycycline treatment. And as we expected in this cluster, the Runx3 transgene was highly expressed. And our favorite genes, the granzymes, were also highly expressed. So these results also confirm that probably the Runx3 can directly contribute for the granzyme gene expression. But we found some additional interesting and putative target gene of the Runx3. So this analysis was performed with the Illumina technology, but as it was mentioned in the previous presentation, that some converting kit is available. So in the future, we also think about to using the MGI sequencer because the reagent cost is lower, and we can get deeper sequencing results. So to summarize the data, we analyzed the very same RNA samples and compared using the Illumina versus the MGI technology. And in case of the MGI samples, we reanalyzed the comparing the hot MPS versus the standard MPS. And as you can see that we found similar and overlapping Runx3 and Zbtb46 regulated gene sets detecting both technology, or even if you use the hot MPS, we got the similar results. So these results will demonstrate that DNBSEQ system combined with the hot MPS is suitable for global messenger RNA transcript analysis. And we also found that and identified some potentially novel Runx3 regulated genes like the granzymes. So finally, I would like to thank to my colleagues who participate in this work, and especially I would like to emphasize the role of Szilárd Póliska, who carried out most of the library preparation, and also he performed most of the NGS analysis. Thank you for your attention. Thanks.
Thank you, Professor István, for a wonderful talk and data sharing. If any would have a question. Yes, Professor Hans.
You obviously have a very good agreement between the different techniques, but you have a huge amount of data, so you can look, or has anybody looked into if the differences are, first of all, bigger than just simply sample duplicates using the same technology, and second, if there's any correlation between the most variable data sets and, for example, GC content or any other parameter. It's a huge difference if you say you have 99% identity, if the 1% are absolutely randomly distributed, or if the 1% happen to be your most GC-rich transcript. So it's quite easy to find out if how random the remaining differences are.
Yeah, actually, we haven't performed a technical replication, but obviously we also try to identify transcripts which are differently expressed. For instance, comparing the hot MPS versus the standard MPS, or Illumina versus the MGI. And actually, so for instance, as you remember, we found around 1,000 genes which were induced or reduced. So within this list, we also compared the MGI versus the Illumina, and we found that 10 out of the 1,000 which show significant differences. But all of them were, it's called kind of pseudogenes. And and probably those transcripts just represent single exons. And we also tried, but we haven't finished this yet, to check the GC content or some other parameters, but we failed to find any correlation. And also we compared the Illumina versus the MGI technology regardless of the DOX effect. And we found around 100 genes showed a significantly different expression. But those looks like also, I didn't show a kind of randomly distributed pattern. And once again, most of them were pseudogenes. So in my opinion, even if we randomly make some other library and run, probably we can get similar differences.
Pseudogenes might very well be most sensitive to slight differences in the sample prep. For example, contamination by DNA. So it might be that you find the same set of genes most different if you simply repeat the same experiment twice. So that's an interesting contour to do.
Okay, thank you.